Welcome. You're tuning in because you want to stay informed, and today we're really diving into something important, a big shift in how antibody therapies are patented. These critical medicines.
Yeah, it's a significant change. For a long time, the approach was, well, pretty broad. You could claim an antibody based mostly on what it did, its function.
Right, claim it by function. But then came this major U.S. court case, Amgen versus Sanofi. And that really turned things on their head, didn't it?
It absolutely did. It basically invalidated those broad functional claims. The court said they just weren't specific enough.
Not going to cut it anymore.
Exactly. That ruling, it sort of drew a line in the sand. It told everyone, look, just saying your antibody hits a target isn't enough for a strong patent. You needed more detail.
More specifics. Okay, so the Amgen v. Sanofi case really pushed things from that broad function to needing precise structural definitions.
That's the core takeaway. You need to define the structure, and specifically the focus has shifted towards the paratope for getting that robust protection.
Okay, the paratope. So if you can't just claim any antibody that does X, Y, or Z, what's the new playbook? It sounds like it's all about claims based on the antibody's actual structure, down to this paratope thing.
That's exactly right. That's the emerging strategy.
And that's our mission for this deep dive, to really unpack this new structural approach, figure out why it's catching on, and how it might offer more solid ground for protecting these innovations. We're drawing on a recent article in Nature Biotechnology, and also insights from Absilico that seem to be right at the forefront of this.
It's interesting, isn't it? It sort of mirrors how small molecule drugs have always been patented based on their exact chemical structure.
Good point.
And now that same level of, let's say, structural granularity is becoming key for antibodies, too. It's like moving from patenting any key that opens this specific lock to patenting the precise cuts and shape of this particular key.
Okay, I like that analogy. Let's dig into this paratope-centric approach then. When we talk about focusing on the paratope, what exactly does that involve?
So the paratope, fundamentally, is the specific set of amino acid residues on the antibody itself, the part that physically touches the target antigen, or the epitope on the target.
Like the fingertips of the antibody making direct contact.
Exactly like that. It's the direct point of interaction.
Right.
So the strategy is all about zeroing in on that physical structure, the part responsible for the binding.
Okay, so you define that contact region, then what?
Go. Well, then the idea is to identify not just the paratope itself, but also what are called permissible substitutions. These are small changes you can make, particularly within the CDRs.
The complementarity-determining regions,
Right.
where the paratope usually sits.
Precisely. You figure out which changes within those CDRs don't wreck the binding, and by defining the core paratope structure in these allowed variations, your patent can cover a whole genus, a family of structurally related antibodies that all work.
A family, okay, so not just one specific sequence, but a structural blueprint almost, with some defined flexibility.
You got it. Like patenting the design for a specific type of wrench head, covering minor variations in the handle maybe, but focusing on the part that actually grips the bolt.
And this structure-based claim, it's considered more robust now, more defensible, legally speaking, especially after Amgen v. Sanofi.
Yes, much more so. Because it's tied to the tangible physical reality of how the antibody binds, it addresses the court's demand for more specific description and enablement.
Makes sense. But, I mean, figuring out the exact paratope and which substitutions are okay, that sounds complex. How do companies actually do that efficiently?
Well, that's where technology comes in, and specifically companies like Absilico. They're developing AI-driven tools designed precisely for this task, building these structure-based genus claims.
So using AI to tackle this complexity.
Exactly. They offer computational ways to do what traditionally involved a lot of time and resources in the wet lab.
Right, saving time and money. You mentioned they have specific tools, Mabtope and MabMature.
Yes, those are the key ones in this context.
Let's start with Mabtope. What role does it play?
Abtope is essentially a computational predictor for the paratope. Its job is to figure out, using computer modeling, which amino acids on the antibody are most likely doing that direct binding to the target antigen.
So it replaces or at least reduces the need for laborious lab experiments like crystallography or mutagenesis mapping.
To a large extent, yes. It provides a powerful in silico, meaning computational alternative. It saves a huge amount of experimental effort.
Okay, how does it work? What's the AI doing?
Basically, Abtope uses AI to build a 3D model of the antibody. Then it simulates countless ways that antibody might interact or dock with the target antigen. We're talking millions of potential binding poses.
Billions. Wow.
Yeah, and then it uses sophisticated scoring functions algorithms to rank those poses, identifying the ones that are most energetically stable and likely to actually occur.
Like a massive virtual matchmaking process to find the best fit.
That's a good way to put it. And the crucial output is identifying the antibody residues that consistently show up at that binding interface in the top scoring poses.
So the ones that keep appearing in the best matches are predicted to be the paratope.
Exactly. That gives you your predicted paratope, a clear insight into the binding mechanism, and importantly, that structural core you need for the patent claim.
And this mabtope method, it's scientifically validated.
Oh, yes. Its development and testing are documented in peer-reviewed journals, the Journal of Immunology and MABS, for instance, so it's built on solid scientific footing.
Good to know. Okay, so mabtope helps you find the core binding site, the paratope. What about those permissible substitutions? That sounds like the job for the other tool, mabmature.
Correct. mabmature picks up where mabtope leaves off, in a way. Once you know the likely paratope residues, mabmature helps figure out what changes you can make in the surrounding CDRs without losing the antibody's function specifically.
Yeah.
Its binding affinity.
So tests out mutations computationally.
Precisely. It systematically analyzes the effect of swapping out amino acids within the CDRs. It predicts, in silico, how each change would likely impact the antibody's ability to bind its target. Will it bind stronger, weaker, or about the same?
And this prediction is key for meeting those patent requirements we talked about. Enablement and written description.
Yes, directly. To get a patent, you need to enable someone skilled in the art to make and use your invention, and you need a written description that clearly shows you were in possession of the invention you're claiming.
Right.
mabmature helps provide that detailed structural information and the predicted functional consequences of variations, which strengthens the case that you've met those requirements for a whole genus of antibodies, not just one.
So the aim is crafting claims that are broad enough to be valuable, covering a family of antibodies, but specific enough, structurally defined enough, to satisfy the patent office and the courts.
That's the balancing act. And mabmature's ability to computationally predict the binding affinity of thousands, even millions of variants is crucial here.
It must let researchers explore a much, much wider range of possibilities compared to making and testing everything physically in the lab.
Oh, absolutely. It's a massive expansion of the sequence space you can investigate. mabmature can really guide the optimization process, maybe even finding mutations that improve binding, while also flagging changes that would likely kill the activity. It cuts down enormously on the required lab work.
And how does it make these predictions? Is it just looking at the sequence or the structure too?
It's sophisticated. It uses a combination of approaches. It looks at both sequence-based information, sort of like language models analyzing the grammar of proteins, and structure-based methods to assess the physical and chemical fit between the epitope and the potentially modified paratope.
Okay, considering shape and charge and all that.
Right. How well do they complement each other, even with those substitutions? That's the question it tries to answer computationally.
So if we were to quickly sum up the advantages of this mab-silico computational approach, what are the headlines? Accuracy, speed, cost.
Those are definitely the big three. The AI tools aim for reliable, accurate predictions for defining these genus claims. Doing it computationally is dramatically faster than traditional methods.
Accelerating R&D and patent filing.
Exactly. And by minimizing the reliance on often very expensive and time-consuming wet lab experiments, you get significant cost savings.
And tying this back to the bigger picture, the core message from that Nature Biotechnology article in mab-silico's work seems to be that the Amgen ruling essentially forced this shift towards structure.
That's really the crux of it. The legal ground shifted. Broad functional claims became much riskier, if not impossible, to defend in many cases.
So this paratope-centric, structure-based strategy isn't just clever, it's almost become a necessity.
It's rapidly becoming the standard, yes. And tools like those from mab-silico are empowering companies to actually execute this strategy effectively. They provide a way to get that necessary structural precision.
Helping them secure stronger, more defensible IP for their antibody discoveries.
Which ultimately is what fuels continued innovation and the development of new therapies in this incredibly important field.
Okay, so let's try and tie this all together for you, the listener. We've seen this fundamental shift in antibody patenting, driven by that Amgen v. Sanofi case, moving away from broad function toward specific structure.
Right, focusing right in on the antibody's binding site.
And the strategy now involves defining Not just one antibody, but a whole family or genus based on that core paratope structure and certain allowed variations, those permissible substitutions.
And companies like Mapsilico are stepping in with AI tools like MabTope and MabMature to make figuring all this out computationally feasible.
Making it more accurate, much faster, and more cost-effective than relying purely on lab work.
And as we noted, it's not just about the legal side. This structural focus also pushes for a deeper scientific understanding of how these antibodies actually work at a molecular level.
Absolutely. So here's something to maybe chew on after this. If these AI-driven approaches make defining and patenting antibodies more precise and potentially faster and cheaper, how might that ultimately affect the pace of new antibody drug development?
Yeah, and who gets access to these treatments? Does it speed things up for everyone, or does it potentially create new hurdles?
Interesting questions. It certainly makes you think about the broader role of AI and computational tools in shaping intellectual property.
Especially in biotech.
It's a space that's evolving incredibly quickly. Lots to watch.
Indeed. Well, thanks for taking this deep dive with us today into this fascinating intersection of law, science, and technology.
A pleasure.
Until next time.